why is it that we can cure chronic pain in mice, like almost every single day of the week, but those exact same seemingly miraculous drugs just fail completely when they're finally tested in humans?
That's a massive problem, yeah.
Right. Welcome to today's deep dive. Usually when we talk about a medical diagnosis, there's this underlying expectation of precision.
Oh, absolutely. Like engineering.
Exactly. Like you break your arm, the x-ray shows that jagged white line, and the doctor just points and says, there it is, that's the problem. But when it comes to chronic pain or debilitating itch, or even just how we feel a subtle breeze on our skin, that metaphorical x-ray machine has essentially been broken.
Yeah, completely broken.
For decades, we've relied on mouse models to understand human sensation, and frankly, it's led pharmaceutical research down these incredibly expensive blind alleys. So today, our mission is to look at how science is finally fixing that.
It's a huge paradigm shift.
It really is. We are exploring a groundbreaking paper from Nature Neuroscience that uses deep single-soma RNA sequencing to map the human touch and pain system. And it's at a resolution we have literally never seen before.
It is, quite simply, the first true atlas of human somatosensory neurons.
Right.
And it's a map we have desperately needed, Pain and itch are, well, they're the absolute definition of diagnostic muddy waters.
Yeah, for sure.
What's so fascinating about this research is how it clears that mud by refusing to rely on proxies anymore. But before we get into the sheer technological leap required to build this map, we need to locate exactly where the sensory processing is happening in your body.
Okay. Set the scene for us.
Right. So we are talking about the DRG, or the dorsal root ganglion.
The DRG.
Yep. Imagine your spinal cord is this massive, high-speed fiber optic trunk cable, right? Running straight down your back.
Okay, got it.
The DRGs are the critical sensory relay stations. They're like junction boxes sitting just outside that main spinal trunk line.
Oh, I see.
So every single physical sensation you experience, from your neck down a mosquito bite, touching a hot stove, the weight of a blanket, a cramped muscle, it all has to physically pass through these DRG junction boxes before your brain even knows it happened.
So if these junction boxes are the gateway to literally all physical sensation, mapping them seems like it should have been step one in neurology.
You'd think so, yeah.
Which brings up the obvious question. To understand why those human pain drugs have been failing, we first have to understand why nobody's been able to map these human junction boxes until right now.
Right, and it wasn't for lack of trying, I can tell you that.
It was because of the sheer technical mountain of sequencing human neurons, right?
Exactly, it's a logistical nightmare. So the researchers' goal was to perform single-cell RNA sequencing.
Let's define that real quick.
Sure. So if your DNA is the master reference library locked safely inside a cell's vault, the RNA is the active photocopy of the specific pages the cell is currently reading to do its daily job.
Okay, so if we can read those RNA photocopies, we know exactly what kind of neuron we're looking at.
Precisely. We know its exact function. But in adult humans, getting that pristine RNA picture from a DRG presents three massive hurdles.
Okay, hit me with them.
First, the human DRG is densely packed with something called satellite glia. These are non-neuronal support cells that physically wrap around the neurons to protect them.
Right, and in standard bulk sequencing, where you essentially grind up the tissue into like a genetic smoothie, those glial cells act like thick acoustic foam.
That's a great way to put it.
Because there's so many of them, their genetic signature completely masks the faint RNA signal of the actual neuron you were trying to study. It's like trying to listen to a whisper in a room full of screaming toddlers.
Yeah, the glial noise just completely drowns out the neuronal signal.
Okay, hurdle two.
The second hurdle is the physical size of human DRG neuronal cell bodies, which are called somata. Right. They are massive compared to mouse cells and incredibly fragile. When scientists try to use standard enzymatic digestion or, centrifuge forces to sort them out of the tissue, these giant human cells just pop.
Oh, wow.
Yeah, they burst like overfilled water balloons, and that immediately destroys the delicate RNA inside.
That's frustrating. And the third hurdle?
Well, to get around that popping issue, previous teams tried a workaround called single nucleus sequencing. They'd just extract the tough, rigid inner core of the cell, the nucleus, and sequence that.
But the nucleus doesn't hold all the active RNA, does it?
Exactly. You miss the full transcriptomic picture because a massive amount of the actively translating RNA is floating out in the wider cell body, the cytoplasm.
Right. So standard methods are basically like tossing a whole library into a wood chipper and trying to read the shredded pages to understand one specific bit.
Perfect analogy, yeah.
But this team bypassed all of that. They used a technique called LCM, laser capture microdissection, combined with something called SMART-C2. So that is the equivalent of taking a tiny, incredibly precise laser and cutting out exactly the one pristine book you want, cover to cover, without disturbing the rest of the library.
It's an incredibly elegant solution to a brute force problem.
I do have to push back here, though.
Okay, go for it.
Anyone who has ever left a bag of spinach in the freezer knows that freezing organic tissue creates ice crystals that completely shred cellular membranes. And my understanding is this tissue has to be frozen, right?
It does, yes.
So how do you snap freeze a delicate cell, shoot it with a literal laser, and not end up with an unreadable genetic soup anyway?
That is the exact problem that stumped researchers for years. But the methodology here is brilliantly protective. The tissue isn't just tossed in a freezer, it's snap frozen almost instantaneously using specialized cryoprotectants.
So that stops the ice crystals.
Exactly. It prevents those jagged ice crystals from forming. It halts all RNA degradation the millisecond it freezes.
That's amazing.
And the laser isn't like blasting the cell. It's used as a microscopic scalpel to trace cut the exact outer perimeter of the cell membrane. It isolates the individual soma completely, lifting it away without any of the stretching or crushing forces of a centrifuge.
It's molecular surgery.
Truly.
And the payoff is staggering. They successfully captured 1,066 completely pristine human neurons. And by applying that SMART-SIG2 deep sequencing, they detected a massive average of about 9,000 unique genes actively expressing per neuron.
To put that in perspective, 9,000 active genes is an unprecedented level of high-definition detail for human tissue. Previous methods gave us a blurry Polaroid. This gave the researchers a 4K 3D map of the cell's entire active machinery.
Okay, so let's do a quick recap and summary of what we've covered so far.
Oh, cool.
Bullet 1, human DRGs are the junction boxes of sensation, but they're incredibly hard to sequence because the cells are huge and fragile. Bullet 2, standard methods just destroy them or miss the data. And bullet 3, this team used snap freezing and laser microdissection to cut out intact cells, letting them read 9,000 genes per neuron in stunning 4K detail.
Spot on.
So for you listening, think about the last time you learned a complex skill. How much did having the exact right tool completely change your perspective on what was possible? Because that's what LCM did here.
It totally shifted the perspective, and with that massive data set, they mapped out an entirely new landscape, discovering what we can call the 16 tribes of touch.
The 16 tribes, I love that. So the data cleanly organized into 16 distinct clusters of neurons.
Yes, 16 distinct tribes within the human DRG. To make sense of this new map, we need to look at how these tribes are broadly divided by two main features, their physical anatomy and their internal chemistry.
Let's start with anatomy, specifically the size and the insulation of the nerve fibers.
Good place to start.
The structural differences here are critical. The easiest way to visualize the two main anatomical splits is by looking at the wiring. First, we have the A fibers. For you listening, think of these as the autobahn of your nervous system. A for autobahn.
I like that mnemonic.
These are large diameter neurons, and they are heavily myelinated. Myelin is essentially thick rubber insulation on a wire. It prevents the electrical signal from leaking out, allowing the impulse to travel incredibly fast. A fibers are built for high-speed, urgent data transmission.
Very urgent.
Then on the other side, you have your C fibers. These are the winding country roads. C for cruising. They're small diameter neurons and completely unmyelinated. Because they lack that insulation, the electrical signal travels much, much slower.
A perfect distinction. Autobahn versus cruising. And the RNA sequencing actually revealed the structural blueprints for these two different roads.
Oh, really? What did they find?
They found a genetic marker called NEFH, which stands for neurofilament heavy chain.
NEFH.
Right. It acts like heavy industrial rebar, and it is highly active in A fibers to support their massive size.
That makes sense.
Conversely, they found a marker called INA, alpha-internexin, that acts as a lighter, smaller structural rebar that exclusively marks the smaller C fibers.
Okay, so that's the anatomy, but chemistry is the other half of the story, right?
Exactly. Chemically, the map splits these neurons again into PE and NP categories.
TP standing for peptidergic.
Yes.
These are neurons that express specific marker genes like CAL-ACA, which produces a peptide heavily involved in localized inflammation and pain signaling.
Yep, CLCA is a big one.
And NTRK1, which is a nerve growth factor receptor.
Spot on. And NT stands for non-peptidergic, meaning they completely lack those specific inflammatory markers.
Okay.
By layering these chemical and anatomical splits, the researchers started uncovering the master control mechanisms. For instance, they isolated a gene called PRDM12.
PRDM12.
Right. This is the master switch. It's a transcriptional regulator that is absolutely critical for the development of almost all of your pain-sensing C fibers. If a cell has PRDM12 active, you know immediately it's in the business of sensing pain or temperature.
The map also defines a few specific functional receptors that we should clarify because they become major players as we go. They identified LTMRs, which are low-threshold mechanoreceptors.
Yes.
Low threshold simply means it takes very little physical force to trigger them. A soft breeze, the light brush of a cotton shirt.
Exactly.
They also mapped TRPM8, which is your primary cold temperature sensor.
Sure.
And they mapped proper, short for proprioceptors.
Receptors.
Those are the specialized fibers that tell your brain exactly where your limbs and body parts are in 3D space, without you ever having to visually look at them.
What's so revealing about this section of the paper is how clearly these 16 tribes are defined by their genetic barcodes. The human DRG isn't just a messy, random soup of sensory cells. It is a highly organized, deeply segregated system. Each neuron type has a specific genetic identity dictating its exact job.
So let's do another quick 5-minute recap.
Yeah.
Bullet 1. We have 16 distinct human DRG neuron tribes.
Bullet 2.
They are categorized by size, the Autobahn A fibers and the cruising C fibers.
Right.
And bullet 3. They're also categorized by chemistry, peptidergic versus non-peptidergic. Each has specific genetic markers, like PRDM12 for pain.
Exactly.
So for you listening, if you had to guess, which type of fiber do you think fires when you stub your toe versus when you feel a soft breeze?
Oh, that's a good question to think about.
Right. If you stub your toe, you feel that initial lightning-fast, sharp pain. That's the Autobahn A fibers screaming to your brain. Then a few seconds later, that deep, slow, throbbing ache sets in. That is the uninsulated C fibers taking the country roads to deliver the secondary pain signal.
It's incredibly elegant how that works.
It really is. But the real shock comes when you take this pristine genetic map of human neurons and overlay it onto the mouse map we've been relying on. Suddenly you see exactly why those billions of dollars in drug trials failed.
Yes. This is where the divergence is massive. And it starts with something as agonizing as a chronic itch.
Let's talk about the cross-species divergence.
Right. This directly answers our opening paradox. The researchers used probabilistic neural networks, advanced machine learning algorithms, to rigorously compare the genetic profiles of these human DRG neurons with previously mapped neurons from macaque, monkeys, and mice.
And what did the algorithms find?
They looked for evolutionary conservation versus divergence. The results showed that things like proprioception, knowing where your limbs are, and basic light touch are highly conserved across all 3 species.
Makes sense. A mouse needs to know exactly where its paws are in space just as much as the human does.
Exactly, just to survive. But the pathways for pain and itch, they have evolved wildly differently.
The divergence in the itch pathway is the smoking gun of this entire paper, honestly. In humans, the sequencing revealed 2 specific non-peptidergic clusters, called HNP1 and HNP2.
The H standing for human, of course.
Right. These clusters almost exclusively express a gene called MRGPRX1. That is our primary foundational itch receptor. If you feel an itch, that gene is firing. But mice do not possess MRGPRX1. They use completely different, unrelated genes, primarily one called MIGPRA3, to sense an itch.
And that is the crux of the issue.
For decades, pharmaceutical companies designed highly targeted drugs to block MIGPRA3 in mice. And in a lab setting, it looks like a miracle. It completely cures the mouse's itch. But when that drug moves to clinical trials and enters a human body, it does absolutely nothing. Because the target it was designed to hit simply doesn't exist in our genetics.
It's the biological equivalent of trying to unlock a door with a key to a completely different house.
Wow, yes.
And the divergence continues into inflammatory itch as well. The researchers found another human cluster, HSST.
Right.
That one responds heavily to localized itch and inflammation. It's marked by high expressions of the SST gene combined with GFRA3 and CCK. It represents a highly specialized, distinctly primate inflammatory pathway.
So it's not just basic itch, it's inflammatory itch too.
Exactly. But the evolutionary divergence isn't limited to just how we itch. It fundamentally changes how we perceive temperature too.
Oh man, let's talk about the cold. because here's where it gets really interesting.
It's wild.
The map revealed a quirk here that explains a truly bizarre phenomenon. Cluster 1 in the human map is defined by hDRPM8. TRPM8 is universally known as the menthol receptor.
Right, the minty feeling.
It's the chemical sensor that menthol triggers, which is exactly why your mouth feels physically cold when you chew mint gum or use mouthwash. But the researchers noticed something fascinating in the human RNA. A significant subset of these human cold receptors also heavily co-express a gene called TRPV1.
Yes.
Now, TRPV1 is the capsaicin receptor. It is the primary sensor triggered by nauseous, damaging heat, like touching a hot stove or eating a habanero pepper.
So just think about the profound neurological confusion this creates. Why would a cold sensor also have a heat sensor built directly into the exact same cell membrane?
Right.
This genetic overlap provides a brilliant physiological explanation for the phenomenon of burning cold. When you touch something dangerously cold, it can activate both the TRPM8 cold receptors and the TRPV1 heat receptors on the exact same nerve fiber.
For you listening, think about the last time you grabbed a block of dry ice or plunged your hand into freezing water. You didn't just feel cold. Your brain registered an agonizing burning sensation.
Exactly.
For years, we thought that was, well, scientists assumed it was just a processing glitch in the brain, like the central nervous system was just getting overwhelmed. But looking at this new single-cell map, the glitch isn't in the brain at all. It's hardwired right into the peripheral nerve itself.
Yeah, the wire sends a mixed signal. And critically, mice do not show this exact same overlapping genetic profile. It is an evolutionary adaptation.
So it's time for another quick recap. Bullet 1. Itch and pain receptors differ vastly between humans and mice. Bullet 2. Human itch relies on MRGPRX1, which mice don't even have.
Right.
And bullet 3. Human cold is tracked by TRPM8, which sometimes overlaps with heat sensors, causing that burning cold feeling.
A great summary.
So, audience, how does knowing that your cold and heat sensors can literally overlap change how you view your body's alarm system? It's kind of mind-blowing.
It functions as a highly tuned dual-thread alarm system for extreme environments. And this theme of specialized adaptation leads us directly into, honestly, the most profound discoveries in the paper.
Yes, let's go there.
Because if our itch and cold pathways are merely divergent from mice, the mechanisms we use to sense deep internal pressure and fast, sharp pain are entirely uniquely human.
The uniquely human experience. This is where the deep single-cell sequencing found clusters that simply have no equivalent in a mouse. They discovered a cluster they named HPEP.piezo.
Let's break that nomenclature down for a second.
Yeah, let's spell it out. The H is for human, PEP is for peptidergic, and piezo stands for a massive, exceptionally high expression of the piezo2 gene.
Piezo2, right.
Piezo2 is a mechanical pressure sensor. Imagine it like a tiny trapdoor in the cell membrane. When the cell membrane physically stretches or pulls, the piezo2 trapdoor pops open, allowing ions to flood in and fire an electrical signal.
The presence of this cluster is a massive deal because of what it co-expresses. Alongside piezo2, these cells express massive amounts of ADRA2C.
Okay, what does that do?
That's a gene intimately involved in the innervation and control of blood vessels. And they also express GPR68, which is a receptor specifically tuned to sense blood flow and shear forces inside your veins.
Wow.
So when you combine massive stretch receptors with blood vessel innervation, what you are looking at is a highly specialized, dedicated cellular system just for sensing internal organ stretch and systemic blood pressure.
That's literal interoception, the biological ability to feel your own internal state.
Exactly.
And it makes perfect evolutionary sense why humans possess this dense, specialized cluster and mice don't. Humans are large, bipedal primates. We stand upright.
Yes, gravity is a factor here.
Exactly. The sheer gravitational physics of maintaining and sensing blood pressure from your feet all the way up to your brain requires a sophisticated, dedicated cardiovascular monitoring system. A tiny quadrupedal mouse running close to the ground simply doesn't face those exact gravitational demands.
Precisely. We evolved a dedicated sensory tribe just to manage our upright existence. And we see this extreme specialization again when we look at how humans process fast, mechanical pain.
Right.
The researchers identified another cluster called HPEP.Cutry. In a mouse, the KIT gene is somewhat generic. It's kind of scattered everywhere across multiple different pain and touch fibers without much exclusivity.
But in the human atlas, KIT is intensely concentrated. It's heavily segregated into one specific A fiber cluster. These are fast-conducting mechanonociceptors.
Right. Nociceptor meaning pain receptor and mechano meaning physical force.
This uniquely human fiber isn't tuned for temperature or chemical burns. It is tuned specifically for sharp, sudden, piercing mechanical force, like stepping on a nail, getting your hair violently pulled, or being pinched. It sends a lightning-fast, segregated warning signal to the brain, completely bypassing the slow burning pathways.
So we have an interoception cluster mapping our internal blood pressure and a dedicated fast pain cluster warning us of immediate physical trauma.
Let's recap this fascinating section. Bullet 1. Humans have a unique HPEP piezo cluster that mice don't have. Bullet 2. It's likely used for sensing blood pressure because we walk upright. And bullet 3. We also have a unique HPEP.KT cluster tuned for fast, sharp mechanical pain.
Perfect.
So for you listening, when you feel your pulse pounding when you're nervous, what if you were actively relying on these specific, uniquely primate piezo2 receptors? Just a crazy thought.
It really is. But as revolutionary as this RNA data is, genetics in a petri dish is still just a proxy. It's a blueprint.
Right. You have to prove it.
The ultimate test of this paper is whether they could prove these microscopic sensors actually perform these specific jobs in a living, breathing human body.
And their validation techniques were just as cutting edge as the sequencing itself. To prove this atlas wasn't just some mathematical illusion caused by the laser cutting process, they first used an algorithm and imaging tool called 10x Enium spatial transcriptomics.
Spatial transcriptomics is brilliant. Instead of extracting and grinding up cells to read their RNA, Xenium takes an intact, highly preserved slice of human DRG tissue.
Okay.
It then uses targeted fluorescent probes to light up specific RNA transcripts right where they physically sit in three-dimensional space.
I love this. It's like moving from a dry spreadsheet of home addresses to a live, glowing Google Earth overlay of the actual neighborhood. You can visually see exactly where everyone lives.
That's exactly what it's like.
The Xenium mapping definitively proved that these 16 clusters physically exist side by side in the human tissue, grouped exactly as the sequencing predicted.
Yep.
But the researchers didn't stop at visual proof. They took it a massive step further into functional clinical evidence using a technique called microneurography.
This is where molecular biology meets live physiology.
The visceral reality of this validation blows my mind. They literally took microscopic needles and inserted them directly into the peripheral nerves of living, fully awake human volunteers.
Awake volunteers, yes.
Yeah. By doing this, they were able to record the individual electrical spikes of single neurons firing in real time while they applied different physical stimuli to the volunteer's skin.
And the results were a perfect vindication of the genetic map.
How so?
Well, let's look at those ALTMRs, the low-threshold mechanoreceptors built to sense a soft breeze. The RNA predicted they should only respond to light touch. During the microneurography, when the scientist lightly brushed the volunteer's skin with a soft tool, the needle recorded rapid, intense electrical firing.
The nerve was screaming touch.
Exactly. But when the scientists applied noxious heat or freezing cold to that exact same patch of skin, dead silence, the nerve completely ignored it.
Wow. It functioned 100% as its RNA barcode dictated. The genetics matched the physical reality perfectly, and this flawless validation brings us to the absolute holy grail of this research, circling all the way back to the failure of those mouse drugs.
Yes, the drug targets.
Why do human trials fail? Let's look at opioid receptors, the primary pharmacological target for severe pain management. In mice, the different types of opioid receptors are mixed and scattered across various pain and itch fibers. It's a genetic jumble.
It is a mess in mice, yeah. But when you look at the human atlas, the architecture is beautifully, cleanly separated. The OPRM1 gene, which encodes the mu-opioid receptor, the specific receptor targeted by powerful painkillers like morphine or fentanyl, is heavily, specifically enriched exclusively in our peptidergic pain clusters.
Exclusively in the pain clusters. Meanwhile, the OPRD1 gene, which encodes the delta opioid receptor, is almost completely segregated away from the pain clusters. It is specifically isolated in our HNP1 and HNP2 itch clusters.
The pharmacological implications of this clean segregation are massive. It means that in a human, if you administer a highly targeted mu-opioid drug, you are precisely hitting the pain pathways without inherently triggering the itch pathways. Conversely, if you want to stop chronic, debilitating itch, which is a massive quality of life issue for millions of patients, you can design a drug strictly targeting the delta opioid receptor. The map tells you exactly where that drug will land and what it will do in a human being.
And mice simply do not have this clean evolutionary separation.
No, they don't.
That is exactly why A drug that works perfectly in a mouse's jumbled system causes completely unexpected side effects, or totally fails to trigger the intended cascade when put into a human.
We finally have the right map for the right territory.
Okay, one final quick recap before we wrap up.
Okay.
Galley 1, xenium spatial mapping, proved these cell types physically exist in human tissue. Bullet 2, microneurography proved they function exactly as predicted in live, awake humans.
Yep.
And bullet 3, human-specific opioid receptor mapping offers new, highly precise targets for anti-itch and pain drugs. So for you listening, knowing that itch and pain have entirely separate opioid pathways in humans, how optimistic are you about the future of non-addictive targeted painkillers?
I'm incredibly optimistic, honestly.
If we step back and look at the sheer distance we've covered today, it's breathtaking. We started with the immense logistical hurdle of laser-cutting fragile, single human cells to bypass that acoustic foam of our biology.
Right.
We explored the 16 distinct tribes of human touch. We looked at the heavy rebar of the Autobahn A fibers and the uninsulated country roads of our C fibers. We unpacked the evolutionary divergence that explains why touching dry ice causes a burning sensation. We discovered the uniquely primate stretch receptors that allow us to feel our own pulse. And ultimately, we proved that this genetic architecture isn't just theory, it is actively pulsing in awake human nerves.
The foundational takeaway here is that medical knowledge is only truly valuable when it is accurate to the specific organism you are trying to treat.
Yeah.
By bravely stopping our historical over-reliance on mouse models, and developing the tools to look directly at the staggering complexity of human genetics, science has opened an entirely new, viable frontier in treating chronic pain and itch.
We started today by asking why those drugs fail. And we found the answer hidden deep in the unique, segregated architecture of our own cells. But it leaves you with an incredible thought to chew on.
Oh, yeah.
If our fundamental biological experience of something as basic as pain, internal pressure, and itch is so intricately tailored, so vastly different from the mammals we share this planet with, what other basic emotions, psychological states, or physical sensations are we experiencing in a totally uniquely human way that we simply haven't built the tools to map yet?
That is a fascinating question.
Right. The map is just starting to unfold. Keep questioning, keep learning, and we'll see you on the next deep dive.